this video we will discuss about this E-cadherin is finally been found out that is the structural component of Invadopodia. So how they come up to this point? So in the previous results we have understood that the E-cadherin and actin's complex formation plays an important role in the Invadopodia formations. So continue uh, continue towards that discussions. What it is, they wanted to check how the Invadopedias, how this E-cadherins and actins plays a role towards this Invadopedia formations. So they stain this Invadopedia and actins along with gelatins at different time intervals, and they categorize them as initiation and the maturation. So initiations, we can see that the E-cadherins are formed and the actins are also started to formed around the cells, and here the gelatin is not been break down. But after some time what they have seen is that the maturation occurs and the number of cells that have this actin and E-cadherin rings goes down. That is, degradation starts, and we can see that thing in the degradations of the gelatins. And here what the researchers are able to see that in the maturation step, by analyzing steps this at step 2 and step 3, they are able to see that the E-cadherin rings are present along with this actin rings, but the and E-cadherin rings alone also presents, but the actin ring alone are not present, so indicating that the E-cadherin ring formation occurs first and then actin ring formation occurs. So in the second case we can see that the degradation begins and the number of cells that have both E-cadherin and actin rings goes down. And in the step 3 what they are able to see that the complete degradation occurs, and that will cause this, the cells will either have the actins E-cadherin positive cells or E-cadherin negative cells with the actin cells that can be seen in this image. This E-cadherin rings is seems to be degraded, and the actin rings also shrink down. And now in order to validate the formation of actins precedes the formation of E-cadherin precedes the actin formations, they did the staining at different time intervals and at different locations, and using the confocal microscopy to find out the depth of this rings. And what they are able to see that in some of the images, that is in the 1.1 images which is shown in this panel E also, they are able to see that the actin ring formations and the E-cadherin ring formations are not in the same same panel. That is the E-cadherin's depth is less compared to the actins. More actins are present in more depth uh, in the Invadopedia. These things are shown using this ABCD points that are highlighted in the 1.1, and the E-cadherin's levels are also shown in the 1.32. And when they did the quantifications in the panel D, we can see that the depth of the actins is low compared to the case depth of the actins is high, or we can say that from the bottom it is low but from the top it is high compared to the E-cadherins. E-cadherins depths are low. So this all together, along with this confocal images of the panel E, which also validate the quantifications that E-cadherins are present at a lower depth compared to the actins. So this all indicates that there is a actins and E-cadherins ring formation occurs, and although they do not form at the same panel, but both of them plays an important role. Or we can say E-cadherin plays an important role, as E-cadherins ring formation occurs first and then actin ring formation begins. So from that they conclude that E-cadherin plays an important role and has a structural is a structural component of the Invadopodia, which is formed from the pancreatic cancer cells. So because in this case they are studying the pancreatic cancer cells in Invadopodia formations. So I hope you understood how they come up to this point that E-cadherin is the structural component of Invivopedia. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.